Hey, Sister Mayo. My group met earlier today, but I wasn't able to join them when they met. However, I can show you that I spoke with Michaela and we went over the answers and stuff. I guess I can shoot you an email with a screenshot of the phone call. Anyways, I'm gonna go over the answers for the group problems. I discussed, like I said, Michaela and I went over them together. So the very first one, what you asked us was, you wanted us to go through the different combination of different vocabulary that we had to learn for question one. So you asked the example of regulatory proteins. So the regulatory proteins are repressors and activators, the negative and positive control. The B, small effector molecules, that's your allolactase, that's your inducers, and that's your CAMP molecules. CAMP activates the cap, which binds the cap site, and then your allolactase will essentially remove the repressor from the operator on the lac operon. Then number C, letter C, which of the terms describes is an example of a sequence found within DNA? So that's your operator, that's your lac A, lac Z, +1 and -10. +1 and -10 are part of the promoter region. And then which above are not part of the lac operon? You got your activator, inhibitor, allolactates, CAMP, inducer, and CAP. And then E, we put lac A and lac Z because those are structural genes. They're genes that encode some protein that is not regulatory. So just those two, just the beta galactosidase, I think is what it's called, and the protease or the promease or whatever it's called, the one that is in the cell membrane. Then you go to number 2. The lac operon is regulated by 2 inducers, allolactase and CAMP. Allolactase, like I said earlier, induces the repressor to move, and the CAMP induces CAP to bind to the cap site. And then B is a lac promoter and the cap site. C is adenylyl cyclase. D is the lac promoter. E is lac L and lac Y. F is attenuation, right? No, attenuation. That's what it's called. Yep. And then G is the merozygote strain. Number 3. Let's see here. What you asked us was whether or not the E. coli cells are going to turn blue when they're metabolized by beta galactosidase. And the X-gal is a molecule that is a substrate for beta galactosidase and essentially metabolize it and turn the whole thing blue. So obviously, if glucose is present, you're not going to be able to do that, because there's the glucose will inhibit CAMP from being produced, and you're not going to be able to transcribe anything. So you can't create the X-gal because you can't metabolize the X-gal because beta galactosidase isn't being made, because you can't bind to the cap, which can't bind to the cap site, which inhibits transcription on the lac operon. Then you go to A. So A, that one is there's no blue because IPTG cannot remove the repressor because it's not there. Beta, or B, excuse me, is blue is there. Yes, it's going to turn blue because the protein to remove the repressor is there. Glucose is absent. Those therefore we're able to do it because CAMP is able to be made by adenylyl cyclase, and they can make the CAMP. And then the X-gal is present, so the X-gal will be metabolized by the galactosidase that we transcribe. And then C, you can't get blue because the X-gal isn't there, so galactosidase has nothing to metabolize. Then D and E are both, you can't have blue. One because, well, first because glucose is present, therefore CAMP is not made. Now there was a little thing on the sheet I remember reading over here. It talks about how when you do have lactose and glucose, you can have a little bit of things being transcribed with some of those structural genes being made. However, in this class, I guess for this purpose, we're saying that the lac operand is off when glucose and lactose are there. So that's my understanding. And then same with E, glucose is present, so it's not going to be made. All right, and then you move on to 4. 4 was fun. Not really. A is inducible if lactose is present. B is non-inducible. C is non-inducible. D is inducible if lactose is present. And then you got E is inducible as well. The reason why E is inducible is because when you look at the IS, that one can't bind, allolactose can't bind to the repressor and remove it, so the chromosome will not be inducible at all. But if you go to the plasmid on this one, the OCs that affect our operator sequence, so the repressor can't bind there, therefore RNA polymerase, or I think it's RNA polymerase, will bind to the promoter region and transcribe if lactose is present. Or excuse me, we don't need lactose, so it's going to be inducible. Or would it be constitutive? No, inducible, right? I'm going with inducible. That's what I'm going with. Okay, and then you're going to go down to 5. Now, the way I set this up may be a little different than what you were asking for. However, what I did here is, for example, for these last 3, C, D, and E, this is the chromosome, and this is the, what do you call it? The plasmid, the F plasmid. So if the chromosome could, has to do with turning yellow. What turns it yellow? I forgot. It's the beta-galactosidase is what turns it yellow and interacts with some other molecule. So what I put is if this, for example, this chromosome will interact when lactose is there. So if the mixture is going to turn yellow, whether or not lactose is there or whether it's not. So that's what makes sense. Yes. So for A, I got with or without lactose, you will not have it's going to be clear. Reason being is you can't make beta-galactosidase. Go down to B, both with and without lactose, you're going to be able to transcribe that structural gene. The reason being is your operator region is free, so the repressor is mutated, so your repressor can't bind. Then you go to C. The chromosome can make it with lactase because the repressor is there, but if lactose is there, then you can just bind to the repressor and remove it, and then the RNA polymerase can bind to the promoter region and transcribe. And then you go for without lactose, you can't because there's nothing to move the repressor. And then for the plasmid, you can do it for both because the operator region is ¿cómo se dice? It's mutated. And then you go to D. The chromosome will make it with or without lactase because the inhibitor isn't being made. And the plasmid will not make it because the inhibitor is being made and the promoter region is mutated, so the RNA polymerase can't bind, and yada, yada, yada. And then you go to E. You look at E. The chromosome will be able to make it because the operator region and the promoter region are fine, and there's no ¿cómo se llama? There's no lac L operand to make the repressor to bind to the operator. There we go. And then the plasmid can't make it because why? Oh yeah, because the lac repressor can't bind to the operator. Or excuse me, the allolactose cannot bind to the repressor on the operator, so therefore the repressor will stay there and nothing will be made. No, like I said, I can send you a screenshot of the phone call, the amount of time I spoke to Michaela about this. I was going to meet with them, but I couldn't. So hope this is okay. But yeah, that's what I did, and those are my answers. Hope you have a great evening, Sister Mayo.